So getting started then, our goal in designing the IonFlux system was to raise the bar in terms of performance of automated patch clamp, as well as lower the cost. And we've made significant progress in achieving these design imperatives with the systems that we offer. In terms of raising the bar for performance, our microfluidic design allows for continuous flow during the experiment and exceptional fluid exchange times. As well, we are able to incorporate temperature control, allowing for automated patch clamp experiments that can be run at physiological temperature. With regard to lowering the price, we have hit an important milestone with the IonFlux 16 system, which sells for a price comparable to a manual patch clamp rig. For those of you who are not familiar with Fluxion, we are a privately held, venture-backed company that was founded in 2005 out of technology developed at the University of California, Berkeley's Sensor and Actuator Center. The company currently has 30 employees with 4 products commercialized and selling globally. We've been awarded 5 NIH Small Business Research grants, including Phase 2 development grants. As well, we have been a 2-time R&D 100 award winner, winning awards for both the BioFlux and IonFlux platforms. We have 2 product lines, the BioFlux systems, which are used for studying the impact of flow and cell-to-cell interactions, as well as the subject of today's webinar, the IonFlux systems for automated patch clamp, which include the IonFlux 16 and the IonFlux HT. Shown on this table is a partial list of our customer base for both the BioFlux and IonFlux platforms. We know how to deliver innovative products to the marketplace, and we know how to support them both in terms of training as well as manufacturing the consumables on a large scale to meet the demand. At Fluxion, we are raising the bar for performance while at the same time we are lowering the cost for performing automated patch clamp electrophysiology. For the throughput levels achieved, we have the lowest automated systems both in terms of hardware cost and ongoing maintenance cost. Simultaneously, we raise the bar for performance by offering the only automated systems that have continuous flow during the experiment, as well as very fast solution switching times due to our unique microfluidic design. As well, our unique plate design allows us to bring a heating element in contact with the bottom of the plate, allowing experiments to be performed at physiological temperature. Additionally, the training time required to get your lab personnel up to data acquisition speed is greatly reduced due to the automated patching process. This allows for greater consistency in the data being produced. And finally, the space required to achieve this is a small fraction of that occupied by other types of automated electrophysiology systems or even an additional manual patch rig. The key applications for the IonFlux systems are those where recombinant cell lines can be used and where a large data set is desired. Chief amongst these types of activity is compound profiling, the key application that has been driving the adoption of patch clamp automation in the pharmaceutical industry for the better part of the past decade. With an IonFlux 16 system, one could screen up to 64 compounds at a single concentration or produce 8 8-point IC50 curves in a single run, and 4 times that with an IonFlux HT system. Alternatively, the system could be used for screening clones, allowing up to 8 clones, or 32 in the case of an HT system, to be compared in parallel. Additionally, one could use this model for comparing the impact of mutations on ion channel expression and function. Taking a closer look at the benchmark of the manual patch clamp experiment. Shown is a typical example of a manual patch clamp rig consisting of a microscope for visualizing the cell to be patched, manipulators for positioning the patch pipette and perfusion lines, and the perfusion system made from syringes, all contained within a Faraday cage. Not shown are the patch clamp amplifier, digitizer, data acquisition computer, and accessories like a bath temperature controller, an oscilloscope, signal conditioning hardware, and or valve controllers. Now, the great advantage of the manual approach is the overall flexibility, allowing either adherent or suspension cells to be used, as well as mixed populations of cells where one can identify the cell of interest by visual inspection. One can do either conventional whole cell patching or inside-out patching or even detach patches. Adding to this flexibility is the ability to change the nature of the experiment on the fly. For instance, if one has a particularly good cell, one may elect to do additional biophysics or pharmacological characterization. It is unlikely that patch automation will ever rival this overall flexibility. The manual technique, however, does require a significant amount of art to master and make productive, and even the best of patchers can usually only do a dozen or so cells in a day, and this level of productivity is far too low and far too expensive to support meaningful compound screening in medicinal chemistry. Hence, about a decade ago, a number of companies began to explore ways of automating the patch clamp experiment. And just for a bit of history, in the 1940s, Kenneth Cole and colleagues developed the modern voltage clamp technique. This made possible the seminal work of Hodgkin and Huxley using the squid giant axon to define the events of an action potential in the 1950s. As well, the work of Hans Ussing was taking place during this time period. During the latter part of the 1950s, Leo Sternbach, who was a chemist working for Hoffmann-La Roche, discovered the forerunner to Valium. The 70s, besides a decade's contribution to music and fashion, was a particularly exciting decade for those of us interested in ion channels and the tools used to study them. First, Singer and Nicholson bring together key work from the 60s to articulate the fluid mosaic model for the plasma membrane of the cell, making possible our modern notion of the ion channel as a protein complex floating in a lipid bilayer. Meanwhile, over in Russia, Kristall and Potapchiko were developing the forerunner of the planar patch clamp technique. Later in the 70s, Sockman and Nair demonstrate that it is possible to record single-channel opening and closing events. And ion channels remain one of the few protein classes that can be studied at the single molecular level in their active state. Also in the 70s, it was found that the Valium binding site was part of the GABA receptor. So nearly 20 years after the discovery of the benzodiazepines, their mechanism of action was beginning to be elucidated. Fast forwarding past the Yuppie and Gen X generations, we arrive at the 2000s, which saw the commercialization of the first automated patch clamp platforms starting in 2002. To be sure, these platforms have greatly increased the interest in pharmaceutical companies for identifying ion channels as tractable targets for drug discovery initiatives by lowering the cost and, equally important, improving turnaround times for compound screening and profiling activities. One of the major drawbacks to these systems has been the high cost of adopting the hardware, as well as the high expense of maintaining such systems. At Fluxion, we have achieved a major generational change in automated patch clamp electrophysiology by employing microfluidics, our lateral trapping technology, and an overall modular system design, which has allowed us to produce an instrument with much lower initial hardware costs as well as lower ongoing maintenance costs, while maintaining the key objective in automating the patch clamp process: increased productivity and ease of use. Given the small footprint of the system, it easily fits into labs where space is at a premium. The systems have only 2 moving parts, so they are much more reliable and easier to work with than alternative instruments commercially available. Moreover, an IonFlux 16 can be upgraded to an IonFlux HT, allowing one to preserve the value of their capital investment in automated electrophysiology when even higher throughput is needed. So how do the IonFlux systems perform the patch clamp experiment? The next few slides will cover the principles of operation. First, we employ ensemble recording, which increases the success rate and reduces variability. Shown is what we refer to as a trap zone. The trap zone consists of 20 closely spaced microchannels in parallel. During the trapping process, 20 cells will be drawn into these microchannels and then taken to conventional whole cell recording conditions using pressure. The inside of the trap contains the internal recording solution, and the main channel with the cells contains the external solution. The entire ensemble is under voltage clamp conditions, such that there is a single measurement of ion flow across the entire collection of 20 cells. The solutions that will flow past the ensemble are connected via microfluidic channels to the main channel. The top of the plate, where the solutions will be loaded either manually or by a liquid dispensing robot, has a standard SBS format, a 96-well format for the IonFlux 16, or a 384-well format for the IonFlux HT. Using a standard SBS format makes it easy for the system to be integrated with standard lab robotics for plate handling and liquid dispensing tasks. Shown on the left is an IonFlux plate, in this case having a 384-well format, ready to be loaded into the IonFlux plate reader. Bonded to the bottom of this standard plate is a proprietary polymer material containing the microfluidic channels. Shown on the right is a close-up view of the microchannels, which have been loaded with dye for visualization in this example. We refer to the collection of chambers and their channels for a set of recording trap zones as an experimental pattern. Shown here is an alternate view of the bottom of a 384-well plate, highlighting the 32 experimental patterns. Each of the experimental patterns is highlighted with orange dye. A plate for the IonFlux 16 would have 8 such patterns. Below is a blown-up schematic of one of the experimental patterns showing the plumbing connections and the relationship to the wells above. Taking a closer look at the experimental pattern, we see that an experiment will utilize 12 wells, 1/8 of an IonFlux 16 plate or 1/32 of an IonFlux HT plate. 2 of the wells, noted with T1 and T2, will be loaded with internal buffer. The T in this case stands for trap zone. So each pattern has 2 trap zones, an N of 2 for each experiment, if you will. There is a single in-well which will contain the cells and the extracellular buffer. The connection of the in-well to the 2 trap zones constitutes the main flow channel. There are then 8 wells, denoted C1 to C8, that will contain the extracellular buffer along with compounds to be assayed, although these could just as easily be solutions with different ionic compositions or other such variations on the theme. As well, one could load a pattern with different internal solutions in T1 and T2 to compare the effect of different internal components. For each pattern, there is a single outwell into which the solutions will flow. All of these solutions, including the cell suspension, would be loaded into the plate, either manually or by a liquid dispensing robot, prior to loading the plate into the IonFlux reader. Once the plate has been loaded with solution, it is drawn into the IonFlux reader and a manifold with the recording electrodes and the pneumatic control lines is mated to the plate, shown schematically on this slide. The control of the pneumatic pressure is defined in the IonFlux control software and allows for fast control for the compounds to be applied in any order desired. Since the fluid flow is all driven by pneumatics, there is no need for robotic liquid handling at this point, making the reader a much simpler and easier to maintain device. The silver-silver chloride electrodes are connected to the amplifiers. Since both the IonFlux 16 and IonFlux HT are using standard SBS format plates, the only difference between the two platforms is the configuration of the manifold, thus allowing for an easy upgrade path between the two. So the process for performing an experiment consists of loading the plate with solutions and then loading the plate into the reader. The manifold then mates to the plate, bringing the electrodes into solution. The pneumatic interface enables highly parallel experiments that are run across the full plate at the same time. Now, this cannot be achieved on automated systems that use robotic fluidic handlers. Beyond the simplicity, one of the nice features of this design is that the bottom of the plate is completely accessible during the recording process. Having this access allows us to bring in a heating element into contact with the bottom of the plate for performing experiments at physiological temperature. The IonFlux systems are the only commercially available automated patch clamp systems which allow for this type of access. So we're truly raising the bar for performance while dramatically lowering the cost of access. Taking a closer look at the perfusion of the cells being recorded. Shown schematically are the two trap zones of an experimental pattern along with the main flow channel containing the cells. Off to the left are the 8 incoming compound lines highlighted with different colors. Solutions are prevented from leaking into the main channel by maintaining a bit of back pressure, which, in combination with the continuous flow in the main channel, holds the compounds in check until the line is pressurized. So at this point, the compound rapidly flushes over the cell ensembles, which in this example activates a GABA receptor, giving rise to an inward current. Once the flow is stopped, the continual flow of the main chamber flushes over the cell ensembles and washes the ligand out of the system, and so on through the series. The solutions may be applied serially with no intervening wash steps if desired, and can be applied in any order and as many times as one desires. As noted earlier, the wells of the plate may be loaded manually, or alternatively, they can be loaded using a liquid handling robot. Shown is an IonFlux HT being used with the TCAN Freedom Evo liquid handling system as implemented in the Fluxion test lab. Any liquid handling robot, such as those from Hamilton, Thermo, or Aurora, could be used with the IonFlux reader. And up to 4 readers can be configured to operate with the liquid handling robot for very high throughput applications. In this configuration, the reader operates as a slave device, and once the liquid handling robot loads a plate into the reader, the experimental process is then initiated, and the IonFlux takes over execution of the experiment as defined in the system software. The system software contains all of the features necessary for defining the experiment to be executed. Shown is the first tab of the experimental setup dialog, and this is where one will define the contents of the compound wells. Within the experimental sequence tab, one defines the pressures that will be used during the priming process, the pressures that will be used during the trapping process, and the pressures that will be used to take the cells into the whole cell recording condition. And finally, the business end of the experiment, the voltage protocols that will be applied, and the sequence of compound addition events. Highlighted is the compound addition editor, showing in this instance a sequence of 4 compounds that will be added in rapid succession, followed by a flush, and then 2 more repetitions of this sequence. Note that the compound application sequence will be the same for all of the experimental patterns. However, each pattern may have different compounds. While the experiment is running, one can view the progress of the experiment. Shown on the right are step responses of the GABA receptor to increasing concentrations of GABA. In this case, the cells are continuously held at a fixed potential, and as the channels open, an inward current of increasing amplitude is seen as the concentration of a GABA is applied for 2 seconds. Due to the pneumatic control and microfluidic channel design, the IonFlux platforms are the only automated patch platforms that can do this kind of experiment. On systems which use a dispensing robot, the robot must return to the compound well for the next concentration to be applied, and then return to the recording chamber to add the compound. This cycle cannot be achieved within 2 seconds, and certainly not across the entire array of recording chambers. During the course of the sweeps, measurements are being made at the points shown with the green and yellow cursors, and the difference currents is plotted in the left-hand window. The difference current measurement has this sawtooth appearance due to the fact that it's capturing the peak current at the yellow cursor during the sweep. Then, in the intervening sweep, the cells are once again seeing the extracellular buffer without GABA, so the current returns to 0, followed by the next application sequence. Now you're saying, but there are only 3 sets of compound applications defined, but there are more than 3 teeth in this sawtooth pattern. This is another unique feature of the IonFlux platform. The experiment can be suspended and reinitiated during the run. As with the manual patch clamp experiment, this is particularly useful when one has a particularly good run and the decision is made that it would be nice to get additional information from the experiment. While it would be difficult to change out the solution in the compound wells, it is possible to change the sequence of additions or add additional biophysics such as an IV plot to the experimental procedure. There are a number of other features such as the amplifier control, temperature control, that we don't have time to highlight in this section. The key IonFlux specifications are that we are using an ensemble recording such that each measurement is from a collection of 20 cells. We are using loose patch technology where the average shield resistance for a given cell is in the neighborhood of 200 megaohm, although higher resistances have been obtained. We have very fast compound on times due to our microfluidic design. Throughput rates vary depending on the specific of the experiments, but if one had a 15-minute long experiment, it would be possible to generate more than 1,500 data points with an IonFlux HT platform. The amplifiers used contain the conventional circuitry for series resistance and leak correction, as well as correction of the fast and slow capacitive components. Additional platform specifications are as follows. There are 2 versions, the IonFlux 16 and the IonFlux HT. The system can be run using manual loading of the plates, but they are also compatible with commercially available liquid handling robots. The instruments have a plate reader format. The IonFlux 16 has 16 amplifiers, and the IonFlux HT has 64 amplifiers. Plate temperature control is available, which allows for running experiments at physiological temperature. And the system comes with integrated control and analysis software. The IonFlux 16 has the lowest hardware cost of any automated system capable of doing more than 1 recording at a time. And as well, the IonFlux 16 could be upgraded to an IonFlux HT. Moving on to some applications of the IonFlux technology and validation of the system. Shown is a partial list containing a number of channels that we have worked with at Fluxion. The recording column shows that we have been able to measure characteristic currents from these different cell lines. The pharmacology column indicates lines where we have done enough work to obtain a more complete pharmacological characterization that is consistent with the literature values. And the optimized column shows target systems that we have worked with enough to obtain success rates in excess of 80%. Note that in some instances, such as the trip channel cell lines, we've had very little working time with these cell lines. And for the most part, our activities have been driven by customer interest, and we're continually updating this collection of data and producing app notes, which are available at the website. Shown on this slide are some of the biophysical properties of the hERG potassium channel, as measured on the IonFlux platform. The hERG potassium channel is a channel of particular interest to the pharmaceutical industry, since the channel has been identified as the primary target involved in drug-induced sudden cardiac death, leading to the market withdrawal of drugs like Seldane and Propulsid. Measurement of hERG activity in the presence of compounds in development is the only in vitro data required for a new drug application being submitted. Importantly, since we are using ensemble and loose seal approach, the fact that these biophysical properties correspond well with the expectation from the literature using the manual patch clamp technique indicates that the IonFlux system has proper voltage control. Equally important, since we are using microfluidic channels to deliver the compounds to the recording ensembles, is the fact that a battery of well-characterized blockers show their characteristic block when tested on an IonFlux system. And even sticky compounds such as terfenadine, which has a high log p-value, are not heavily skewed. Shown on the table are a set of reference compounds, their log p-values, where log p is a measure of the octanol water partition coefficient and can serve as an indication of stickiness of the compound. The IC50 values as measured using an IonFlux, and then the range of values reported in the literature. Note that for sticky compounds like terfenadine and Bepredil, the literature values vary widely. An important ability of the IonFlux instruments is to rapidly apply and remove compounds to the cell ensembles across the entire plate while the current is being recorded. In a sense, the system is akin to incorporating an 8-channel rapid perfusion system for each recording ensemble. We've looked at the GABA receptor as an example of a ligand-gated ion channel. Our measurement of 3 different GABA agonists agrees well with expectation from the literature. Despite having a 50-year history in the pharmacopoeia, GABA modulators remain a vital research interest in the pharmaceutical industry. As well, research labs continue to elucidate the functional roles of the various GABA receptor subtypes. An understanding of their differential pharmacology will be important for the next generation of GABA modulators. Here we show the results of examining some well-characterized blockers, as well as positive allosteric modulators, such as the diazepams. The values obtained using the IonFlux system agree well with the expectation from literature. Consistency of the experiment is particularly important when one wants to compare investigational compounds to one another. Shown on the scatter plot are the current amplitudes in response to a 10 micromolar GABA stimulus for each ensemble on an IonFlux 16 plate, shown in red, i.e. there's 16 dots for each plate, with 8 plates measured over the course of a day. Extracellular buffer was the negative control, shown with the black dots. The bar graph shows the average current amplitude for the 16 ensembles of a given plate, along with the standard error bars, as well as the Z prime calculation for each plate shown in red. This is a very robust assay when performed on the IonFlux platform, and this consistency means that one can have confidence in results measured in the early day with respect to experiments done later in the day. In this experiment with the nicotinic acetylcholine receptor alpha-1 subtype, each concentration of acetylcholine is applied twice, with interleaved control concentrations also applied twice. Shown in the inset is the consistency of the interleaved control applications for a single ensemble, which gives us confidence in the concentration response profile produced. As we go through escalating concentrations of acetylcholine, we see the characteristic increase in the rate of opening of the nicotinic channels. And this was an experiment performed on a single IonFlux HT plate, so we can see from the N of 62 that we had a very high success rate of 97%, and the concentration response profile agrees well with expectation. Most important for the nicotinic experiment is we have very fast recovery times due to our microfluidic channel design. Shown in the upper left is a trace in which the second application of acetylcholine is applied 1 second after the first application. You can see that the channels have not fully recovered, so the amplitude of the second response is smaller. Shown in the lower trace is a similar experiment where the two pulses of acetylcholine are separated by a 10-second period. After 10 seconds, the second application produces nearly a full response. So then plotted is the recovery time curve. Importantly, in this experiment, we have not added any acetylcholinesterase to the buffer flowing between applications, which would need to be done on other automated platforms, and is even frequently done in the manual experiment. And this is due to our high flow across the cell ensembles, giving a very thorough clearance of the acetylcholine. And this is another example of how we are raising the bar in performance for automated patch clamp experiments. The voltage-dependent sodium channels, or NAVs, are fast voltage-gated channels, and we use them to demonstrate our ability to record fast voltage responses. Shown in panel A are the characteristic current voltage plots for two members of the NAV family, NAV17 and NAV18. In panel B is a lidocaine concentration response experiment for NAV17, and in panel C, a concentration response experiment for NAV18. The concentration response curves are plotted in panel D, and the potency difference for tonic inhibition matches well with the literature. A final example showing the quality of the voltage control using our ensemble approach is KV 2.1. Shown in the upper panel is the current voltage relationship for the voltage-gated potassium channel. Shown in the inset is the manual patch clamp data collected in our test lab, showing excellent correspondence in the IV relationship. And as well, this data was in perfect accord with published data for this channel. To start summing up, the IonFlux systems represent the next generation in the evolution of the automated patch clamp technique. At Fluxion, we are raising the bar for performance while lowering the cost of access to this technology. In terms of raising the bar for performance, our microfluidic design allows for continuous flow during the experiment and exceptional fluid exchange times. We're able to incorporate temperature control, allowing for automated experiments that can be run at physiological temperature. In terms of lowering the price, the IonFlux 16 system sells for a price comparable to a manual patch clamp rig and is the lowest cost automated platform that can make more than one measurement. The IonFlux 16 systems are validated automated patch clamp platforms which will help to accelerate your research by minimizing the time needed for training and increasing your data acquisition capacity and improving the consistency of the results. I'd like to thank you for taking the time today to learn a bit about the IonFlux systems. If you'd like additional information or a system quote, please contact your regional distributor. Thank you.